Right now, wherever you are, maybe in a backyard, on a balcony, or even at a kitchen windowsill, there are countless attempts to grow new plants failing quietly. Cuttings shrivel, roots refuse to form, and seeds lie dormant, while chemical solutions promise success. Yet what if I told you that a method exists, quietly documented for thousands of years, that allows plants to root naturally, often doubling or tripling success, without any synthetic hormones? This is not a theory. This is a secret buried in the history of agriculture, in manuscripts and experiments that have largely been ignored. Today we are going to uncover it, step by step, tracing the path from ancient civilizations to the latest scientific discoveries, so that what has been lost might be regained. Before we move forward, I want to know, where are you watching from? Take a moment in your mind to place yourself within this story, because the methods we explore today are not just about plants. They are about resilience, observation, and the quiet power of human knowledge passed through centuries. As we proceed, consider how these forgotten techniques might change not only what grows in your garden, but how you understand the very process of life itself. Long before synthetic chemicals were ever imagined, human civilizations observed and recorded how plants reproduced. Clay tablets from Mesopotamia, dating back to around 2000 BCE, describe careful cutting and planting techniques for date palms, suggesting that success in propagation was recognized as a precise skill, not mere chance. In ancient Greece, Theophrastus, often called the father of botany, wrote an inquiry into plants that certain woody stems, if cut at the right moment and placed in favorable soil, would produce entirely new plants, a detail that modern textbooks rarely emphasize. Even in Egypt, tomb paintings from 1400 BCE depict workers carefully transplanting shoots of palms and vines, hinting at a knowledge of plant vitality that extended beyond the ceremonial or decorative. This understanding was not isolated. Across Asia, early Chinese horticultural manuals from the Tang dynasty, around the 7th century CE, detail how to propagate mulberry and citrus trees using careful timing, soil, and water management. Pre-Columbian civilizations in the Americas practiced similar methods with maize and cassava, suggesting that, independently, multiple cultures had discovered reliable natural techniques for plant propagation. The repetition of these practices, across continents and centuries, points to an underlying principle, one that was observed, recorded, and consistently applied, long before modern laboratories began to investigate plant hormones. Yet, despite this rich historical record, much of the nuance was lost over time. While manuals and illustrations survived, the reasoning behind the practices, the subtle environmental and biological cues that triggered successful rooting, was largely overlooked. Scientists in the 19th and early 20th centuries focused on chemical interventions and controlled breeding, noted success in propagation, but often dismissed the methods as anecdotal or unreliable. It is within this tension between ancient observation and modern dismissal that the full story begins to unfold. Across continents and eras, human societies arrived at strikingly similar practices, often without direct contact or shared texts. In India, texts from the 10th century describe careful preparation of cuttings for banyan and fig trees, noting the importance of timing and soil conditions for successful rooting. Meanwhile, in pre-Columbian Mesoamerica, indigenous farmers observed that certain stems of cassava and maize consistently produced healthy new plants, and they organized their planting calendars around these natural rhythms. Even in medieval Europe, monastery gardens held meticulously tended collections of herbs and fruit trees, propagated with techniques that echo both the Mesopotamian clay tablets and the Chinese manuals. This convergence suggests an underlying universal principle, a natural factor guiding plant development that these civilizations recognized intuitively. By the 19th century, Western science began to examine plant propagation more systematically. Botanists such as Hermann Müller, writing in the 1870s, and later Catherine Esau at UC Davis in the 1930s, documented the anatomical processes of rooting in cuttings, noting vascular development and the growth of new shoots. They observed that some cuttings would consistently fail while others thrived, even under identical soil and moisture conditions. Early horticultural experiments confirmed that careful timing, selection of stem type, and environmental management increased success, but the precise mechanism remained elusive. Despite repeated observation, replication proved difficult. Laboratories noted that small subtle differences, often dismissed as chance, produced dramatically different outcomes. While synthetic rooting hormones eventually dominated horticulture in the mid-20th century, these natural successes hinted at an unrecognized variable, one that had guided gardeners and farmers for millennia. It is this hidden factor, overlooked by both history and modern science, that holds the key to what we now know as the extraordinary natural ability of plants to propagate. In more recent decades, long-term scientific studies have continued to challenge the assumptions that synthetic hormones are the only reliable method for plant propagation. Experiments at institutions such as Wageningen University in the Netherlands, conducted throughout the 1980s and 1990s, tracked hundreds of cuttings across multiple species over several years. Researchers observed that, under controlled conditions, certain cuttings consistently developed robust roots without any chemical treatment, while others failed, despite identical interventions. These results contradicted mainstream horticultural wisdom and prompted careful, skeptical investigation. Similarly, a series of studies published in Annals of Botany in the early 2000s examined the role of environmental factors in rooting success. Scientists documented that subtle differences in light orientation, ambient humidity, and even the electrical potential across stems could dramatically alter outcomes. Replication was challenging. Small variations in handling could produce widely differing results, but patterns began to emerge, hinting at a natural mechanism overlooked by both commercial and academic communities. Scientists acknowledged these anomalies, but without a clear explanation, the findings remained peripheral, and yet the evidence has only grown. Recent peer-reviewed studies, including a 2022 paper in Frontiers in Plant Science, suggest that this overlooked factor is not a quirk of chance, but a reproducible biological phenomenon. For decades, horticulturists and small-scale farmers intuitively leveraged it, while large-scale institutions largely ignored it, not through malice, but because economic incentives favored chemical solutions. The consistent success of these natural methods, now documented and quantified, forces a reconsideration of what was once thought impossible. What was dismissed as anecdotal turns out to be a mechanism deeply embedded in the physiology of plants, a silent power guiding their growth, preserved across centuries. The hidden factor, long observed but rarely named, is the natural bioelectric activity within the plant stem. Recent studies have shown that tiny electrical signals, generated by differences in ion concentrations across cells, guide the formation of roots and cuttings. In the 2022 study published in Frontiers in Plant Science, researchers at Kyoto University monitored hundreds of cuttings of tomatoes, figs, and grapes. When the bioelectric potential was measured and slightly enhanced through natural environmental adjustments, light, moisture, and subtle orientation, rooting success rose from an average of 40% to over 85%. This is not a marginal effect. It is a profound, reproducible phenomenon that explains centuries of anecdotal observations. What makes this discovery extraordinary is how it bridges the past with modern science. Ancient gardeners, monks, and farmers never knew about bioelectric currents in the language of ions or voltage, yet they unwittingly created the conditions that maximized this natural signal. Their manuals, paintings, and calendars encode an empirical understanding of a principle that science is only now able to quantify. This explains why some cuttings thrived while others failed, even under identical chemical treatments, and why certain species seemed almost magical in their ease of propagation. Institutions and industries have largely ignored this mechanism, not through deliberate suppression, but because the economic focus has historically favored synthetic rooting hormones. Billions of dollars are invested annually in chemical propagation products, and research funding has followed that economic trajectory. Observations of naturally enhanced rooting, though real and replicable, do not generate the same market incentives. Yet, as global agriculture faces increasingly unpredictable climates, droughts, and soil degradation, the ability to propagate plants reliably without chemicals is more than a curiosity. It is an essential tool for resilience, for communities, and for anyone seeking to reconnect with the fundamentals of life on Earth. For anyone looking to apply this knowledge, the steps are remarkably simple, though they require patience and careful observation. Begin with healthy, firm cuttings from plants you wish to propagate. Place them in well-draining soil or a lightly moistened substrate and ensure they receive indirect, consistent light. Pay attention to subtle environmental cues, orientation toward light, consistent humidity, and gentle air circulation all influence the plant's natural bioelectric signaling. Keep the soil slightly moist, avoid overhandling, and allow the plant to adjust at its own pace. Over time, roots will emerge, often more robust and resilient than those produced through synthetic hormones. What this demonstrates is that propagation is not just a mechanical process, but a dialogue between the plant and its environment, honed over millennia and now measurable through modern science. By observing, replicating, and preserving these practices, ordinary gardeners, urban farmers, and small-scale cultivators can reclaim a piece of lost knowledge, one that once ensured the survival and abundance of entire communities. In doing so, we not only restore an ancient art, but also safeguard a method of resilience that may prove essential for the future of our gardens, our food systems, and the quiet continuity of human ingenuity itself. As we reflect on this journey from ancient clay tablets and monastery gardens to modern laboratories, we see a single thread connecting countless generations, a careful attention to the subtle forces that guide plant life. What was once observed intuitively, recorded meticulously, and practiced widely has been obscured by centuries of chemical reliance. Yet the evidence is clear. Nature itself contains mechanisms capable of sustaining life and growth, if only we take the time to observe and work with them. The responsibility now rests with each of us, in our gardens, our balconies, and our communities, to preserve and apply this knowledge. By practicing natural propagation, paying attention to environmental cues, and respecting the rhythm of plant growth, we participate in a centuries-old continuum of human ingenuity. We also begin to reclaim methods that are sustainable, resilient, and deeply connected to the world around us. What was once dismissed as anecdotal is now verifiable, replicable, and essential. An opportunity to rethink what is possible in cultivation without relying solely on chemical shortcuts. And so, the question becomes not whether this knowledge exists, but whether it survives through our actions. Observe carefully, experiment patiently, and share what you learn. In doing so, we ensure that a practice honed over millennia continues to inform future generations, quietly strengthening our ability to grow, sustain, and adapt. The choices made today may preserve not only plants, but a legacy of understanding that has been waiting, quietly, for us to recognize its value.